Okay, so welcome back to E443, 543, our second lecture. Thank you, people, for coming back. For those of you who are back, I appreciate always that people show up. I hope that the homework and things were okay. Some people came to the virtual office hours yesterday. I realized it was a holiday, so it wasn't the best time to have office hours, but at least we had an opportunity to talk for an hour or two with some people. As always, I need to remind people that Engineering 443, 543 is live streamed and recorded. I don't know if anybody's tried looking at the YouTube. I did do a rough edit of the YouTube recording of this. People want to look at material after the fact. So today, we're going to be doing a little bit about using nanoHUB. I realize that some people in the class are using the nanoHUB app, some are not. If you're not using the nanoHUB app, that material won't be that exciting to you, although you can try it out. If you are using the nanoHUB, then you'll need it. And then we will begin to work with Computational and using it. So there was a Homework 0 questionnaire quiz, which asks you a little bit about your background. There are some people who haven't filled it out. If you fill it out, you get an extra 10 points for your homework, so you might as well do it. Again, if you're in the room, please run your Zoom so that with the sound turned off. So that you could do screen share. Again, once we've been through this a few weeks, it'll shake down, I'm sure. So last time we introduced the idea of trying to understand organization of tissues in the context of development, maintenance, called homeostasis, developmental diseases, infectious diseases, toxicology, and bioengineering in medicine. Since cells are the active agents that build and maintain tissues, building models from the cell level upward and downward makes a lot of sense. And we have to understand both the biophysics of what cells are doing and also how they're controlled, which is mainly biochemical. And we introduced this idea of what we call virtual tissues, which are multi-scale models that typically represent control in the form of biochemical networks, cells and their behaviors as agents, so these are agent-based models, and then the extracellular environment. The extracellular environment can be represented at different levels of detail itself. The most common thing to include in the extracellular environment is chemical species, diffusible chemical species, sometimes the mechanics of extracellular matrix, sometimes fluids, depending on what you're modeling. And sometimes there'll be a higher level organization which represents the circulatory system, which is generally not represented in a spatial form. One of the problems we face in biology is that there are a huge range of length scales and time scales. That's not so unusual in physics, but what's a little bit different between biology and physics, probably not that many physicists in the audience here, but if you were a physicist, physics is based on the assumption of homogeneity. In physics, you depend on the fact that every oxygen atom is just like, every oxygen molecule is just like every other oxygen molecule. Every water molecule is like every other water molecule. And that allows you to define things like temperature and viscosity. In biological systems, you have a lack of homogeneity both within scales and between scales. That means that a lot of the fancy mathematics that people do in physics doesn't work. And so things are harder problems. And so you are left with much more ad hoc approximation in biology than you face in physics. In principle, you have biological processes happening on the timescales of milliseconds or microseconds, for example, during metabolism. If you want to understand how electrons move around during metabolism. And of course, we have lifetimes on the scale of years. If you want to understand how cancer develops and progresses, we have to be able to understand how things change in the body over timescales of months or years. And so that range of length scales is quite challenging. Typically, the kinds of simulations we're going to be doing, we're going to be looking at timescales on the order of minutes to months, and that you can handle all in one level of detail. But if you want to go to very fast timescales, things like blood flow in the body, which is fast, then you do face problems in terms of trying to represent everything with one kinds of simulation. There are technical mathematical ways of doing what's called timescale separation. Those work, for example, in neuroscience, but they don't often work in tissue level models because you get what's called overlapping of scales. Those are technical issues which you don't really have to worry about now, but I want to give people a heads up on some of the issues they might face. We looked in the class at some examples of different kinds of simulations that you could do with a package like Computational. We didn't have time last time to go over all of these examples. The top one on the left is a relatively detailed model of a single cell crawling around, generating a actin cortex in the lamellipodium, causing movement. The second simulation was a rather abstract simulation of how evolution happens in a tumor, of somatic evolution in tumors, of how metastasis evolves in tumors. The third simulation was a much more detailed, realistic simulation of the interaction between a growing tumor and blood vessels. The fourth simulation was actually a flow transport model showing how when you take a drug, different parts of the liver are exposed to different concentrations of the drug. Down on that second row, angiogenesis was a model of the invasion of blood vessels in the retina and the destruction of retinal integrity. In the middle, we have a plaque assay model where we've infected cells and we're seeing how the immune system responds to that infection, for example, in the lungs for a disease like influenza or SARS-CoV-2. And then on the right was the simulation that we had of cyst formation in kidney. And so one of the things that we have to face when we're building these kinds of models is all of these simulations, except for that one on the top right, have individual cells as agents in them. But we are dealing with vastly different levels of detail. In the top left, we have a lot of detail in a single cell. The middle row right also has a lot of detail in the individual cells. But in that tumor angiogenesis simulation, we have really quite a few cells, tens of thousands of cells in that simulation. And so we can't afford that much detail. And so one of the things that Computational is pretty good at is allowing us to vary the level of detail with which we represent individual cells. We can represent a lot of cells at low level of detail or a fewer cells at more level. And you see some of these simulations are intrinsically three-dimensional, some are two-dimensional, and some are what we call 2.5-dimensional. That is that they're 2D sheets, but layers of 2D sheets on top of each other interacting. So today I would like people briefly to talk about their possible project ideas. I realize that it's hard probably for some people to do that so early on, but I'd like people to begin to get to know each other. I created that Google Doc for people to try to put in preliminary ideas. So far, only a couple of people have filled it out. I would encourage you to do that as a way of getting together and building coalitions to work on projects. I really would like to see a possible 3-man teams, 3-woman teams 3-person teams to work on that. I'd like to hear a little bit about people's homework experience from the homework that was due today. And then we'll go into a quick review of how to upload and download CompuCell simulations to nanoHUB, for those of you who are using the nanoHUB application. And then we're going to start digging into actually how CompuCell works and see how far we get in terms of how to understand what CompuCell does, the internals of CompuCell, how it represents cells and cell behaviors, and then how to control cells in a CompuCell simulation. In particular, thinking about membrane fluctuations and contact energies as control parameters. And they're going to be on the online version of this, there'll be some extra slides that go into more detail that we won't cover in class today. So before we get too far along, I'd like, if possible, to have people go around the room. And again, this is why we need people to be on Zoom, even if you're in the room, because we to be able to connect, we need you to be able to do that. Why don't we start with people who are online? And it's okay to say you don't have any idea what you want to do with a project. If that's the case, you might just give your general area of research interest or study interest, so that I get an idea, people can get an idea of whether, if they're looking for somebody who's a biologist or a computer scientist or something else, how to build a team. So why don't we start with I'll just go down the list as we see it. Maybe start with Gabriel, if it's okay. And I have to.
Hello, can you hear me all right?
Can people in the room hear him all right?
Is that good? All right. So I'm Gabe. I'm a PhD student in cognitive science with a minor in bioengineering. And so, you know, a lot in, cognitive science is a weird discipline. So you got people working in neuroscience and psychology, and then you got people like me who, you know, think that, you know, like how we understand flight from an airplane, how we build an airplane to understand flight, we might build something cognitive or some sort of artificial life in order to understand cognition, or some of these really interesting things that we see in biology. So yeah, for this project, I'm kind of oscillating. So I'm really, of course, interested in everything to do with synthetic biology and regenerative medicine, but I was thinking of doing something. So I also do some work in the olfactory epithelium. I was thinking about doing something about wound healing or regeneration or neurogenesis in the olfactory epithelium. But I also really like, and I'm really curious to see so a lot of my other work is in evolutionary robotics too. So I would like to see with agent modeling is super fun. And CompuCell I'm really excited to dig into. So very interested to see something like, you know, maybe we can evolve like a locomoting agent, or maybe we can evolve like some sort of, but I'm still deciding is basically the general idea, but something to do with regeneration, something to do with possibly robotics. I don't really know yet.
Okay, great. Thank you. Logan.
Hi, my name's Logan. I'm a PhD student in microbiology with a minor in bioinformatics here at IU. And my projects are pretty biology centered, you know, as is my expertise. So I study quorum sensing. That's my thesis is centered on quorum sensing in bacteria. So how bacterial populations use communication networks to coordinate population level behaviors. And that includes things like infection or bioluminescence or biofilms. And my main interest for the project is one of two things: either modeling how the timing of a quorum sensing inhibitor impacts infection outcomes. So, you know, if I give someone a quorum sensing inhibitor at the beginning of their infection, how does that look versus at the end? And it's very similar to the coronavirus model we looked at, but it's actually quite different because of the way these behaviors are regulated. So that's why I was really excited about that. And then the other one was looking at how the evolutionary emergence of what are called cheaters, so bacteria that listen for quorum sensing but don't contribute to the public good, can cause population collapse during infection in the host.
Well, there definitely are published models of quorum sensing generically, and also of cheating, and the evolutionary selection for cheating or against cheating. I don't know if the people have modeled specifically treatment and its role, the role of quorum sensing in treatment outcomes.
So
But
yeah, I've looked and there aren't, and that's actually part of the reason why I took this class.
So I think that, but to get started with modeling quorum sensing, there definitely are published papers we could use as an initial result. And if Gabriel's interested in collective phenomena and emergence, that's sort of quorum sensing is not a bad example. The evolutionary one is an example. So maybe it's worth talking together a little bit about that. Great. Carmen.
Hi everybody, my name is Carmen. I'm a senior in bioengineering. I'm not really sure exactly what I want to do yet with the project, but I'm definitely most interested in like the cancer therapy or the a couple of the papers that have to do with tumor growth.
So again, I mean, being flexible is not a bad thing when you're building teams. So I hope that this kind of discussion at least gives people an opportunity to begin to talk to each other about that. JH.
Hello, my name is JH. I'm a first year PhD student in molecular neuroscience, and my research interest is in Alzheimer's disease. So I found a paper talking about like how inflammation works in Alzheimer's disease. So like 3 agents work in this situation, like neuron, microglia, and amyloid beta deposition. So how they are interacting each other, but it's quite old model back in 2003. So if I can replicate this, I would be interested in this. So I'm definitely from more of biological background. So I'm looking for someone who is interested in like coding and bioinformatic background.
Great. Okay, so let's see. Elmer. Okay, if you just want to talk here, you can use this computer to do it. But the only problem is going to be if it's trying to be screen share.
Yeah. Okay. Can you hear me, guys? Can they not hear me? Yeah. Okay. So I'm Elmar Bucher, and I started a PhD this summer. I'm a bit older, and I have background in, I would say, in the small version is in bioinformatics. So I'm quite a geek. I know programming. I know the Linux operating system, and on the other hand, I have knowledge from bioinformatics, from cancer research, and from biology. So what I'm leaking is the mathematical background sometimes of modeling and understanding that. That's what I'm trying to catch up here. And that's where as well my direction in the PhD goes. So my major is in bioengineering and my minor is in computer engineering. And for the model that I have chosen, this is based on a paper, and they use a model, a wet lab model where they do organoids, this is called, and it's very, very primitive biology that they look at. So it's just a breast cancer cell line. One time there is ECAD knocked in, and one time there is ECAD not knocked in, and there is an extracellular matrix represented as color chain fibers. And what they study in this paper is more or less the density of this matrix and how this influences proliferation and migration. And they have even provided a model, a physical model, about a continuum model. And my idea is to model this as well with Physicell and to model this as well with Computational 3D. And then I have 3 models that I can compare and see what are there, get we different insights, or what are the different artifacts that the model will provide, and learn so about in general about modeling and what the power is of modeling. That's the idea for my project. Thank you.
Okay, so who do we still need to hear from, please? And maybe easiest just to. We'll figure out a way to get this to work. Up to you, Dave.
Hey guys, I'm Nick. I got my bachelor's in neuroscience. I'm now getting my master's in intelligent systems. So I kind of went from biology and neuroscience, and then I've spent the last maybe 2 or 3 years doing more engineering, computer work, programming type things. The paper I was looking at during the homework when we were talking about projects was the paper on slime molds. I read a few papers about them in the past, just the portrayal of intelligence as such like a simple organism. And I saw there was the simulation that someone did in Computational. That looked really interesting. I don't really have that specific of a project idea. I also thought the project that JH mentioned, because my background's in neuroscience, the whole Alzheimer's disease, glial cell is definitely in my wheelhouse too. So I thought that was really exciting.
You want to try, do you want to come up to the front or do you want to try from your computer? Either way. Well, why don't you just come up to the front for the moment? It's a relatively large class. For everybody will take a little while, but I think it's important that everybody introduce themselves at the beginning of the class. Go ahead.
Hi, everybody. My name is Ibrahim. I'm a first-year PhD student in Intelligent Systems Engineering. I'm doing bioengineering, and I work in Maria's lab, and we do zebrafish research. Specifically, we've been tracking zebrafish embryonic development and their responses to various chemicals. So that's kind of the project realm that I was thinking about, is modeling zebrafish embryonic development. And it's I guess response to different chemical exposures.
Is there a specific part of a specific organ system or specific phase of development that you're working on in Maria? She does a lot of blood vessel development work. You have a focus?
Our project is just we're monitoring twitch response in late stage embryo. I don't know if that's reasonable for Computational, but obviously we can narrow it down to a specific organ system if that's what's needed.
I mean, there's also this big institute, NSF-funded institute at Purdue, which is focused on zebrafish development. So that's potentially, Maria isn't formally a member of that institute, but there's certainly a lot of people at Purdue doing zebrafish development and modeling thereof. And so there might be some possible outreach that you could do for support for your research for that. All right. Let's see. Did you want to try either in person or in front of the? Let me see if I can do it. Oh, okay.
All right. No. Oh.
What if I do that?
All right. No. All right. Now. Testing. Yep. Okay. And you guys can hear me now? Okay, cool. All right. So hello, I'm Mike. I'm master's in environmental engineering. So I didn't really have a set idea, but there was two things I was kind of thinking about. I was also interested in slime molds and just like bacteria colonies and, you know, spore growth and proliferation and just all the kind of interactions that kind of go into that. So that was something I was kind of looking into. And then the other one that I found from like the papers that I found interesting was just like cell differentiation in like embryonic growth and such. So that was just, there was a couple articles that I found interesting with that. But that, yeah, those were the two main ideas that I had in mind.
Okay, great. So let's see if I can unmute now that I've muted. So it looks like there are two mute buttons on this computer, which makes things a little more complicated. But people, not very pretty. Okay, how's this? Is the screen share still working for people online? Okay. Is there anybody who did not speak? Who needs to?
I didn't.
Okay, go ahead. No. Okay, so is there anybody who did not speak?
I have not yet.
Okay, go ahead.
My name is Hayden Moore. I am a nanoscale engineering undergrad. And my interest for this project, I mean, I'm pretty open-ended, but ideally I would like to do something with bone tissue growth and or cartilage formation, which would be in line with my senior capstone project.
Yeah, I think that hearing people's ideas, they're all great ideas. My one concern is that if everyone runs off and does their own thing, it can work, but it's a lot more work for you. And so, if there's a way to find common ground so that you can build teams, I encourage that, because I think it'll make your lives a lot easier. Bone formation and remodeling is a wonderful topic. They're beautiful models, for example, by David Basanta at Moffitt, for example, of the role of bone maintenance mechanisms in metastases to bone. So there's actually quite a strong connection between cancer modeling and bone modeling. There are also some beautiful published agent-based models of bone development and bone remodeling. So these are all great topics. I hope that people will use that discussion document and work together to try to come up with some great teams that can do this. I'd say there could be people who are interested in quorum sensing and collective phenomena. Dictyostelium are great, but bacteria also have collective phenomena that are not so different. So it may be possible to come up with some shared projects. So I'm looking forward to working with people to try to help come up with a couple of good collective projects. Any other questions or comments before we move on? Okay, there were a couple of homework problems. The first homework problem was we did that very quick simulation in class of cell growth and division, and the homework asked you to dig into that a little bit. Is there one person who might like to show some results from that? Is there anybody? Okay, Logan, you want to do that?
Oh no, I just had a excuse me, I had a question about this problem actually.
Go ahead.
The Canvas homework assignment and the Word document had two different sets of instruction for this problem in particular. I followed the one on the Word document, but I just wanted to make sure that was correct.
Yes. So the problem with Canvas is that actually putting things like equations and images into Canvas is a pain. On the other hand, if you're going to upload your homework, I have to create a placeholder assignment in Canvas so that you have a place to put it. There should be in most of the homework assignments, there should be a note saying something to the effect of, don't look at the Canvas assignment, look at the Word document or the PDF. So if there's ever a deviation between the Canvas version and the Word version, please use the one that I emailed to you or the one that's in the student materials folder. And if for some reason somebody can't, I try to create a PDF as well as a Word document for everything, but if I forget to do the PDF, please send me an email reminding me to create the PDF if you have trouble using the Word document, okay? So yes, thank you for the clarification. In general, I'll try to make them similar, but in particular, you'll find that when I upload the PDF, upload to Canvas, some formatting will disappear and equations will disappear. So since the homeworks are generated in Word and the Canvas uses a strange markdown format, which isn't compatible with Word. That's a good point. Okay, does anybody? As I said, class participation is something that is expected here. So at some point, you probably will get called on and say, please show what you got. And if the answer is you didn't get anything, that's okay. But so would somebody like to just show us very quickly what they got for the problem 2? I'm getting a lot, a high rate of lack of enthusiasm here, which is okay. Logan, your question, comment? You willing to do it?
I'll do it. Yeah.
Okay, great. Thank you for jumping in and being willing to do that. So we should have the screen share available to you now. And again, just take a minute or two. I mean, the goal here is not to spend a half an hour on this. It's just to get a sense so that people can get a sense of what people did and how it worked. And if people have questions about a homework assignment, then you could do it together to get feedback.
Can everyone see my screen?
Mhm.
Okay. So I started off with changing the cell division parameter. I played with this a lot during class, so I have no clue if I changed it back to the default volume or not. But when I opened it up from after class, my cell division volume was set to 25, and I decreased that to 5, just by changing the if cell.volume is greater than integer. And what I noticed was that when the cells started off, they started off like the normal size they were, but as they started to divide and the simulation continued, you can kind of see that the cells were smaller than before. I also noticed they formed this contractile ring around the center as they grew out and divided. And I just said that most of these changes were just because I had decreased the cell division volume from the original 25. So before the cells were reaching their target volume, they're dividing again. And that's creating the smaller cells that we see on the screen. And then I also mentioned how it looks like I can see more cells. And again, that's just because I can, because they're smaller. And then I worked with the contact energies or the adhesion energies. And I changed these to be higher. So I made it, I increased them to 10 from 1. So I didn't change the contact energy with the medium, just between all the cell types. And I noticed that what happened this time was that instead of like what was random heterogeneity, I started to notice that there were clumps of the same color cell advancing out from the center mass, and that those clumps were maintained for the duration of the simulation. So you can see here the red corners down here, and then the green corners up here, those were the same from the beginning to the end of the simulation I ran. So this makes sense because I'm increasing cell to cell adhesion. So rather than splitting apart as they divide, they're kind of propelling the mass forward as they spit out cells behind or in front of them, which made sense to me since I decreased the adhesions, or increased the adhesions, excuse me. And then for the last parameter for the growth rate, I increased the growth rate from 10 to 100. And the primary thing I saw here was that the lattice filled way faster than normal. So before it was filling at 1/10 of the rate, and now I was seeing a more explosive growth. And I noticed again a similar contractile ring where there was a center mass of larger cells that slowly got smaller and smaller as the mass expanded.
Okay, great. Does anybody have anything they want to add to that? That's a good lead in for how to present results. As I say, a lot of these problems are pretty open-ended, and so it's normal to have different results from different people. And especially at the beginning when you're exploring how to do these things, what's a reasonable answer will depend a little bit about on where you come from in terms of your background and interests. All right. Did anybody try the problem number 3? I think last year some people had that model crash on nanoHUB. I don't know if people were able to run it. How many people were able to get it to run? All right. Does anybody want to talk briefly about their results? Let's see.
Hey, can you guys hear me all right? I have headphones, so I think it may, I don't think you have to mute yourself. Either way, okay. My Zoom client that I have has echoes a lot, so I'll use my headphones anyway. And I'm sorry I don't have my camera on because it unfortunately does not work right now. I can share my screen. Okay. Can people hear me? Could I see like thumbs up or something? Yeah. Okay. Gabriel's giving me a thumbs up. Okay. Yes. Number 3. Can you guys see my document all right? Yeah. Okay. So for number 3, first changing this K parameter. See if I can zoom in a bit. Changing this K parameter, I let it run for 300 steps in the simulation, and I just saw with a larger K equal to 100 versus equal to 1, my population of uninfected decreased a lot faster and a bit steeper. And then you could also see down here, cells were dying at a much higher rate. They were also releasing virus at a much larger rate. And yeah, so since K changes the virus's affinity to the receptor, it makes sense that my immune response was stronger, I believe, in the K equals 100 case, which would make sense because I had a lot more death, a lot more virus releasing, but it's hard to see too. My immune cell count was starting to increase as well. For the R max, so I tried 1.0 for 300, kind of as a control, and then I did, after 100 epochs, I changed it from 1.0 to 0.1, and then I did one other where I ran it from 1.0, changing it to 0.1 after 300 epochs. And what I saw that was really interesting was in just my control simulation, I started with an increase, and then there was a decrease in the immune response that eventually led to a larger increase as the infection started to spread. But when I changed the R max value after 100 epochs, so here's my 100 if you're having trouble seeing that, instead of having that same increase, it kind of lulled and stopped this return growth of the virus. And then that's even more pronounced if you changed it after 300 epochs is here. So we had a really similar immune response to the control, except at 300, right when it was changed, we had a really steep decrease in the immune cell response, which I took to be in the concentration of the immune cells. So yes, I said here, I believe I'm forgetting exactly what R max changed now, what it said in the homework, but I believe it was directly related to the immune response. So it made sense that I saw such a drastic increase. And then also, since between changing it at 100 epochs and 300, you could see, looking at my death data compared to the control, it's relatively similar. We had a bit of neural cell death, not neural cell, just cell death, and a little bit of infection. And then when we waited till 300 epochs to change the Rmax, we had a lot more cell death, a lot more infection, and then some death actually of viral releasing cells, if I'm interpreting that correctly. And then for this third value, the beta delay, I believe is beta decay. I may have put it incorrectly. Oh no, beta delay. Yes, so that I had 0.1 versus 10.0. A huge difference, mostly in this immune response. Also, in the control is 0.1. You can see we had a really nice secondary immune response that just plain and simple did not happen in the second case. It just kept kind of dwindling. It looked like it was starting to pick up over by 300. So if I ran it longer than 300 epochs, we may have seen a little bit more. And then the death data was totally different. We had in the original control, some virus releasing cells were the ones that were mainly dying, whereas in the typical, or the beta delay equals 10, we were seeing infected cells, uninfected cells, and like actively dying cells. And I believe if they're in the death data plot, and please correct me if I'm wrong, but I believe this is the kind of cell that was dying. So I took that as, with the beta delay down so far, I believe that was directly connected to the immune response as well. Yes, the speed of the immune response. So with a slower immune response, it made sense that we had a lot more healthy cells dying, whereas with a faster immune response, a lot more viral infected cells dying. So yes.
Okay, great. Thank you. So we'll have plenty more opportunities to. Everybody. I hope as time goes on, I think that we'll get smoother at these transitions between in room and out of room. But I want to make sure that everybody can participate, and so we'll work on that. So for the current homework, this homework may be a little bit long, and I want to say two things. One, I'll try not to assign homework problems that are not things that we've covered in class, but it's certainly possible. That sometimes we're going to wind up having homework problems that I've written up that we don't get to in class. We may not quite finish what I planned on in the class. If that's the case, it's perfectly okay for you to say in your homework solutions, I'm going to hold this till next week. Don't feel, it's good. Sometimes I will give homework problems which are a little bit of a challenge, which will say, I'd like you to think about a problem before we cover it in class so that you're motivated to understand why we're covering it in class. But in general, if a problem seems to be unreasonably demanding, feel free to say that. I know you have, this class is time consuming, but I also know you have other things to do. And so don't put your whole life into filling out the homework problems for this class. Don't be too intimidated by that, please. And it's fine to ask for advice to me, Juliana, or Hayden in terms of what was reasonable or what's expected. What I do hope is that everybody gives every problem a good try. But if there's a problem with it, we can discuss it in class and work on it together or work on it separately. That's all right. I wanted to go over homework problem 6 on the homework for this coming week. And this is a bit of an experiment. What I am asking you to do, and you've got 2 weeks to do it, and so you get double credit for it, is, since there are a lot of people here who use PhysiCell or other modeling environments, I'm asking people to look at Computational as much as you've used it so far, and then comment essentially on usability issues and capability issues. And I'd like each person to find 2 other modeling environments. I'm not asking you to go in and run lots of simulations or do lots of in-depth things. I'm talking about a half an hour, 45 minutes worth of level of engagement, unless you really want to look at some of the other packages. And then I've created a Google spreadsheet, which has a list of packages and then a place for you to put your name, and then a set of questions which I'd like you to fill out. Some of these are yes or no. Some of these are more descriptive. I'd like you to say, what do you think about the thing? What are its advantages or disadvantages? What kinds of models does it have available? And of course, everybody in this class is using Computational. So as a sort of a dry run for that, you could answer those questions about Computational. And then what we have is a list of other modeling packages that we know about. And there are actually quite a few other ones besides the ones that we know about. And what I'd like people to do is I'd like everybody to do the exercise for Computational as a dry run. And then for this week, pick one of the other packages and do that. And then you can either do a second one for next week, or from the list that we have, or find one that you found yourself online that you're interested in. Because as I say, there are a lot more. But the one thing that I'm asking people to do is to not all clump on one package. So if you're going to do, I don't know, Artisto as the one you're doing, put your name in that list. And then if you're looking for one to do, pick a different one. Because what I'd like, this actually could be a really useful resource for people. And if we cover all of them. And so I'd like people to try to spread out and have at most two people doing the same package. So think of this as a little bit of a interlock table, where if you say, I'm going to work on Biocell, and put your name in that table, claim it for yourself. And then if you're somebody who comes afterwards, pick a different one. And if all of them are taken once, then by all means go and do a second review of one of the ones that's been done. But try to spread it out. Are there any questions about that? Does that make sense as an assignment? And the list of questions that I've proposed in the spreadsheet are some basic ones that Giuliano, Hayden, and I came up with. If you think there's something you want to add, you could certainly create a column called Other and put any other comments you want in it. But those are the basic ones. And if people have trouble getting access to that spreadsheet, please let me know. But it should be available to people. And again, the point of this is not to spend a week reviewing these things, but do look at it enough that you can get a sense of, is this something that's usable? What's it for? Is it developed for a particular community? I mean, some things are focused on cancer modeling. Some things are focused on neuroscience. Some things are very focused, like Biocellian is a high-performance computing package. Of course, a lot of people use PhysiCell here in this class. Biocellian and PhysiCell are very similar in many ways. And so there are a bunch of these kinds of questions that you could ask. And frankly, it's interesting to us also, if you say, wow, there's this great demonstration or great educational material or the great documentation for package X or the way they present their material online. We didn't really ask about online presentation, but the way they present their material is really good. And those are things that we can try to do to make this class better and our material better too. So that can be helpful. For example, Morpheus has a very, very good model repository. I wish we had for Computational an equivalent model repository like that. And so if anybody has any questions, please come back to us, me, Hayden, or Giuliano about that. But does that give a basic understanding of that homework problem? The other problems in the homework assignment are all pretty much based on what we'll do with the rest of the class. So I think those should be more straightforward. Okay, so I want to spend a few minutes. Can I get a show of hands again? I sent out an email. The email, I had a questionnaire. The questionnaire, I asked people how many people were using Computational locally and how many were using it on nanoHUB. And the majority of people said they were using nanoHUB deployment. And so because of that, we have to deal with a couple of the issues that nanoHUB has. nanoHUB is great in the sense that you don't have to worry about installing software on your local machine. Everybody has the same interface, but it has some limitations. And you've used the nanoHUB installment if you ran that model that we talked about, the viral infection model. But one of the problems that you face with nanoHUB is that file uploads and downloads are not straightforward. We can't upload or download directly to your workspace in nanoHUB from Computational. The other thing is that you can't cut and paste between your desktop and the model editor. So if I type something in Zoom chat, I can't have you cut and paste it into the model editor. You can cut and paste within the models. That works just fine, but it has to do with the fact that X11 is so ancient that it was developed before clipboards were invented. And so the idea that there would be a global clipboard that would be accessible wasn't part of the X11 standard. As I say, the interface that is running Computational is older than many of the people taking this class. So it's not surprising it's a little bit out of date. Computational itself is a lot more modern than that, but the interface that nanoHUB supports, and I think actually some of the Physicell apps also use an X11. Some of them are more modern, but some of the uploaded nanoHUB apps in Physicell also use the X11 interface. So one of the things that we'll use is Jupyter Notebooks, and we'll just use Jupyter Notebooks to do uploads and downloads. It's not that you have to learn any Python or Jupyter or anything else. It's just for uploading and downloading. It also allows you to cut and paste between that and nanoHUB. If you're using the desktop version of Computational, you don't have to do all of this. So let me just walk you through it. Let's do a quick exercise. In the student materials folder, there is a demonstration called Colonic Crypt, and it was actually written by a student in this class as a class project some years ago. It was a student of Paul's, actually, of Paul Macklin's, and it was really quite a beautiful simulation of how the Colonic Crypt works. And you should be able to find it in the student materials directory. And the first thing you have to do, whether you're running it locally or going to run it on nanoHUB, is go to student materials. There should be a folder called demos for project class 2. And inside of that, there should be a zipped file called something like Colonic Crypt. It should say colonic_crypt_cc3d. And you'll need to download that to your local machine. Whether you're going to use nanoHUB or not, you need to have it somewhere you have it accessible on your local machine. And instead of doing a split screen, I can do a split screen if people want, but maybe Giuliano, I can ask you to make sure that this works as we go along. So I'll give people a few minutes to do that. And I've been trying, I don't know if these QR codes are helpful to people, but I've been trying to use these QR codes for things to, so that if you, especially if you're running from home, they may be helpful in terms of not having to go after these long URLs. If people need help getting this to work, please let me know. Giuliano, maybe you could put the link in the chat if you have it as well. So is everybody able to find that demo? Yeah, you got it. As usual, I should probably be asking who has not been able to find the demo rather than who has. You couldn't get it? Right? You still need to download it. Should be. How about our online people? Could I get a thumbs up, thumbs down for whether you were able to download the? Okay, Jonathan, you got it. Carmen, were you able to get it? Yeah, JH, you were able to get it. Comment that if you use Firefox, it'll sync tabs between your phone and your local machine. I think Chrome will do that too if you set it up. Okay. Chrome, you have to enable tab syncing between your applications. Yeah, the QR code sort of assumes that you're using a cell phone, so the QR codes are sort of new to me, but I've noticed in conferences that there are a lot more people who put QR codes in their talks. And I really do find that useful. In other words, if somebody says, I have a article such and such, or I have a simulation software, I will point my phone at their camera at their talk and download the web link, so I don't have to retype everything. If it really doesn't work for you people, then tell me. But it's a bit of an experiment. But I thought, especially if it's recorded, let's say on the YouTube, then having it available later on maybe is not a bad thing. So we'll try it. Does anybody need more time? I'll try the poll. So we've got our poll system, which is basically if you need more time, say you need more time. Okay, everybody seems to have it. I realize that one of the issues with the poll when you're using Zoom is that the poll will always come to the front of your screen, which can be a little bit annoying. So I'll try to not use them all the time, but I want to make sure that people know. So everybody has that downloaded. Good. So now that you've got downloaded, if you're going to use nanoHUB, how many people are using nanoHUB at the moment? Give me a thumbs up if you're using nanoHUB. Is anybody using nanoHUB? JH is using nanoHUB. Anybody else? Okay. Okay, well, for the people who aren't using nanoHUB, I asked your patience for a minute. So what we're going to do is we're going to go to nanoHUB dashboard. And so launch nanoHUB. Go to your dashboard, go to all tools, and type in Jupyter, Jupyter. And then it'll give you a bunch of examples. One of them will just say Jupyter Notebook, and bookmark it because you'll be using it a lot. And even if you're not going to be using nanoHUB as a primary resource, it's not a bad idea to do follow along for the moment. So I'll give people a minute to do that. Any one of them will work. One of the issues you'll face in nanoHUB is that things are forever. And so if they're old versions sitting around, they will stick around. And so I know that the one that I point to gives me a warning that it's an older version, but any one of them will work. Any one of the Jupyter notebooks will work. Okay, so with these polls, if you've got it worked, that's fine. If you need help, let me know. But I don't want to go forward until everybody's got it working. And if you're online and you need help, feel free to unmute and ask for help. And again, for the moment, we're only using Jupyter notebooks to do uploads and downloads to nanoHUB. There's an FTP server and there are a variety of other services that are available, but I find the Jupyter notebooks, a lot of people are used to using Jupyter notebooks for other courses. And so to me, that one is the one that's most likely to be familiar to people. This will be painful for the next 5 minutes, but once you've done it once, it's actually really easy. Just once you figured out the workflow, it's easy, but the first time is a little bit painful. Okay, does everybody got it now? Or are we still, I can see how we're doing here. Does anybody need help? Okay. All right. So once you're in Jupyter notebooks, there is, let's go back a sec. When you launch Jupyter notebooks, you will get a menu that will say Files, and there will be a button that says Upload. So click the button that says Upload. And that will pop up a dialog box, a Finder box, and you should be able to point to the simulation that you just downloaded from the Google Drive. And again, it would be more convenient if we could upload directly from Google Drive to nanoHUB, but it's a security thing. No, upload the zip. So one of the things that nanoHUB does not allow you to do is to upload a file structure. It only allows you to upload single files. So since CompuCell simulations are actually file structured, that you have to do a zip. Okay. And so you find your file and then you have to actually hit the blue upload button to do the upload. Did that work for people? Okay, I see a lot of nodding. Anybody need more time? Okay, so now go back to CompuCell on nanoHUB. So launch your CompuCell app. Sorry? Well, we're going to use it one more time, so I'd leave it in the running. So Jupyter, so nanoHUB lets you have 3 active. Hayden, what's the question? Go ahead.
Oh, I was just saying I need more time. It's not letting me upload the zip.
What's it saying?
It's just the upload button is grayed out.
Is anybody else having that problem? What app are you using to do that? Which nanoHUB app are you using? You want to screen share it? Let's take a look at
Jupyter.
it together.
Yeah. Oh, it won't let me share while you're sharing.
Oh, I can. Go ahead.
Okay, so
Hit upload, yeah.
and then
Okay, so you need to find the zip file. You're inside of the zip file.
okay, it wasn't like this a second ago. What have I done?
Excuse me, I'm teaching the class.
Hayden, you have to like upload. You can't extract the zip file. You have to upload the zipped folder.
Okay. I did not think that I extracted it. Maybe it did it on its own. I'll re-download and try again. All right, thank you.
Okay, were you able to get it to work? Sorry about the interruption. Somebody seems to have sent a bunch of students to this classroom for a ghost class. So I hope they find their classroom. Okay, so are you still stuck or did it work? Sorry about that. I got interrupted. How are we doing?
I think that it's automatically unzipping whenever I download it.
It shouldn't do that. If it is, then you need to zip it again.
Okay.
So rezip it. Are you going to be using nanoHUB or not? You running local?
I'm on nanoHUB.
Okay, so then we have to get this solved. So let's work on that. Other people? Did anybody else have this problem?
Okay, I got it.
You got it? Okay, so if you got it uploaded, that's good. So now we need to go back to Computational. And in Computational now, you should be able to go to Twit. And in Twit, you should be able to go to open file. But perhaps in this case, I should be showing this to you in the split screen. So how's it going? Are people able to get into CompuCell now? Okay, so I'm going to launch CompuCell. Inside of CompuCell, I'm going to go to Twit, start Twit. People seeing this all right on the screen share? Start Twit. I'm going to open a file, open CC3D project, and I go to my root directory, and I want to find one that's zipped. Okay, the ones that I asked you to upload are zipped. Okay. These are almost all okay. So I'll do here angiogenesis. That's not the one you're going to be running. See if I have it here. Colonic Crypt. Zipped. Here we go. Example colonic Crypt. So you open it, it'll tell you that could not create a default directory because it already exists. And I have to put it somewhere. You shouldn't get that error because you should have it. And I can create something like colonic Crypt. I've already got 2, 3, and 4 because I've done this so many times. I'll do 4. Not found. There we go. It will throw an error. It will throw a warning message, but it should actually do it. Let's see. Well, let's see what happens now. It's here. So how did that work for people? I'm not giving you a great example here. I hadn't planned on doing it myself, but if people are having trouble, then I want to make sure that it works. If you did it local, so if you're running CompuCell locally, then you just type Twit in your Finder. So how are people doing? I don't have the viewers. If you give me a thumbs up if you got it to work. 2 thumbs up. 3, 4. Okay, who's stuck? If you're stuck, raise your hand. Okay, how are people doing? Yes, no, yes, everybody. Okay. All right. You get a warning. And now you have to, now that it's unzipped, you have to open it and put it. And so if people maybe have already jumped ahead of me, but you should now be able to open to that file, point to that CC3D file and open it. So in my case, it would be Colonic Crypt, Colonic Crypt, Colonic Crypt. Yeah. Okay. Does that work for people? You got it open? How about our online people? How's it going? 1, 2, 3. Anybody stuck? Need help? Yes, no, your work? Everybody? Okay. Okay, so we've opened it. And now that you've opened it, you want to run it. So let's run that. We'll go here. And we'll say Colonic Crypt. I'll right click on it, open in Player. And then I have to switch the tab if I'm in nanoHUB. And you should see the simulation launch. People get it to run? Now, if you're running locally, you wouldn't have had to go through all that. You would have just downloaded it and opened it in your local Quit and unpacked it locally. So the extra step is the fact that you have to use the Jupyter Notebook to upload it. Okay. Anybody need help or time on that? As I say, I understand that this is not the most convenient thing in the world to begin with, but actually, once you've done it a couple of times, it's about 2 minutes per upload. Right. So now if you've got it running, again, if you're going to switch between Quit and Computational Player in nanoHUB, it's the tab buttons at the bottom. This is the code. This is the running simulation. If you're running locally on your local machine, Quit and Player will be separate applications. You have to use the tabbing system on your Windows or Linux. So that's one of the things that's actually a little more convenient in nanoHUB than it is in the desktop. All right. So why don't people actually try to run with this simulation a little bit? Exactly what this simulation is and what it does is something we could talk about. But it's a simulation of the maintenance of It's a simulation of the maintenance of cells in the gut. So in this particular simulation, there are stem cells at the bottom of the colonic crypt. Those stem cells differentiate based on signals in their environment and lead to the migration of cells upward to the top of the colonic crypt, where they're eventually sloughed off and die. And you can see, for example, there's a chemical field, which is the WNT gradient, which you can visualize here. That's an example of a simulation that you can run. I'm going to skip the visualization exercise for the moment because we're a little bit behind. I'd like people to get to play with Computational code on your own rather than worrying about uploading and downloading. But I do want to quickly show you how to do a download as well. So again, the limitation with nanoHUB is that we can only upload and download single files. And so if we're going to write a simulation on nanoHUB and we want to save it to our local machine, In Twittit, and this is important, in principle, your local directories should be persistent on nanoHUB, but I can't absolutely promise you that they will be there. And so when you write code on nanoHUB, I strongly recommend that before you end a session, you download whatever you did. Obviously, if you're using your local machine, you do it whatever you normally do in terms of backups. I encourage you to version your software a lot. But on nanoHUB, it's important to do a backup. And so let's go to Twittit. And what we'll do is in Twittit, you select the top level list for your simulation. You right click, you say zip it, you save that zip file to your local directory. And let's actually do that. So here's my Colonic Crypt simulation. I right click, I say zip it. I'm going to put that in my home directory. And because I already have done this a million times, I've got to give it a number. I'll call it Colonic Crypt Simulation 5. And that will save it. So the download is actually easier than the upload. So everybody do that save. You'll have to give it a new name because you've already got the base directory name there. Do the save. And once you've done that save, you go back to your Jupyter Notebook instance that's running there. And I actually don't have Jupyter Notebooks running myself, so somebody here could try it. Go back to your Jupyter Notebook. And then within Jupyter Notebook, you have a tick box that you can select for the file, and you can hit download, and that'll pull it to your download directory. So try that out. Make sure you can save and download. And I'll give people a minute to try that out. That's a relatively complicated 3D simulation. One thing you will find with nanoHUB is that sometimes it gives you a lot of CPU time and it's very fast, and sometimes it's slow. It's a public server, and so it depends a lot on how many people are using it. We've benchmarked it. It's usually a little bit slower than running it locally, but on average, it's actually pretty fast. But there are times when it's slow. And so it's like, I mean, if you use Colab, you can get the same thing. Or Google will say, okay, now you get lots of CPU time, and another time they won't give you so much. So those are the breaks when you're using somebody else's compute resources. Occasionally, nanoHUB will not work, and that's a different question. So if nanoHUB doesn't run at all, then we need to know. But normally it's pretty quick. Okay, so does anybody need help with the download, or did that work for people? Looks like people got that pretty well. So that's a little bit clunky, but it is. It's not. I hope it's not too horrendous in terms of what you need to do. In my experience, once you've done it a couple of times, it's pretty efficient. It's never gorgeous, but it's not ruinously expensive to do it. I understand the first time you do it, it's not much fun. Okay. So why don't we take a 5-minute break? This seems like a good time to do that. And we'll be back at 6:25, and then we'll start with the class proper, which is talking about how Computational actually works. So everybody stretch your legs, get some tea or chocolate bar, and I'll see you back here in 5 minutes. If anybody has any questions, or problems that they want troubleshooted while we're in the break, that's fine. Just let me know. Okay. So thank you for your patience. As I say, I hope things will be simpler going forward. But I want to make sure that we take the time to make sure everybody can upload and download things. And now we can actually start doing some Computational. Giuliano, do you want to introduce yourself while we're here?
Hello, everyone. I'm Juliano. I'm going to be your AI for the semester. You've probably seen me post some things in chat already. And I'm organizing some time on my week to set proper regular office hours. I should announce them soon on campus.
Okay, so Giuliano is an expert in multi-cell modeling. You'll see online several papers that he's published using Computational and other modeling methodologies. And so I hope that people are able to go to him for help on all sorts of things. So now in our remaining half of the class, I'd like to talk about the basics of Computational. One can get into a lot of technical detail on these things. This is going to be a fairly high-level overview. There are references if you want more detail, and there also are additional slides, which I'm not going to show, but are in the deck, which will be available to you to dig into some more detail about some of these questions. So is this, just before I go further, is the screen share working okay for people? Make sure everybody sees it all right. Yeah. All right. So the first thing that we have to decide whether we're going to do a simulation of cells, this is going to be true whatever modeling methodology we use, is how we're going to represent cells as individual agents in a simulation. And here's a picture of some cells. These look like epithelial cells, that is, cells in a line, in a sheet of cells in a tissue. We could talk later about the different kinds of organization of cells in tissue. And there are going to be a bunch of slides that I'm going to skip, but that will be available to you in the deck that go into quite a bit more detail on the difference between what are called epithelial and mesenchymal cells, essentially whether the cells have fundamentally two-dimensional or three-dimensional organization. And so one way that we could represent cells is what are called cellular automata, where we have a lattice, and each cell on the lattice represents one cell in the biological system. And that has some advantages. It allows you to write very large simulations with millions of cells. It runs very fast. And a classic example of that could be, for example, NetLogo, which is designed around cellular automata models. Although NetLogo these days is actually quite flexible, allows you to do a lot more than that. Another way that we could represent cells would be what are called particle center models, where the cell is a point, and we represent its interactions with the outside world in the form of a potential. These are based on biochemical simulations we call molecular dynamic simulations. And there are a lot of packages that do this. Physicell is one. Biocellian is another. There are dozens and dozens of packages that use this methodology. It has some real advantages. It's off lattice. You're not confined to having your particles having discrete locations, which is nice. It's fast. Also, you can do large scale simulations, but it does have some limitations. Again, there's no perfect methodology. All methodologies will have some trade-offs. And it's interesting at some point to understand what those are. And one of the things that'll be very nice if we have Physicell people in the room is to sort of understand what those trade-offs are and how they play out when you build simulations. Another way to model things, especially if you're modeling sheets of cells or what are called vertex models, where instead of modeling the cell as an agent, you model actually the corners around the edge of the cell as agents, and you connect them with little springs. And that may seem sort of artificial, but in epithelial sheets, very often the cells have what are called an actin cortex, which is a set of fibers that run just inside the cell membrane, parallel to the cell membrane, and are fairly rigid. And they also are active. They actually are contractile. They pull. And so modeling the cell as a bunch of essentially motors that are contracting along its edges is not a bad way to model things. And that kind of modeling is used especially for modeling fruit flies. There's a whole community that does fruit fly models. And also models of plants, often use vertex models. Those are very efficient in 2D. They're a little more complicated to get working in 3D, although there are some packages that do that. And center models can be extended. For example, Tissue Forge, which is our center model package, is going to support vertex modeling. The other methodology that's commonly used, and there are more than this, there are certainly many more, but these are some of the most common, is the one that we're going to be using in Computational, which is a lattice-based method, but where each cell occupies a lot of lattice sites rather than one. And of course, there's a trade-off. In a cellular automaton, you could have a lot of cells because each lattice site is a cell. Here, if you have every cell have 100 lattice sites, well, you basically get, for the same lattice size, you have 100 fewer cells. So that's a trade-off. The advantage, though, is that you can choose how many voxels to give to each cell, and so you can sort of trade off the resolution of the cells spatially as well. In some ways, this kind of model is the most naive one, because if I imagine an image, a digital image of the cells, I could just go through and say, this voxel in the image is in cell number 1, and I give that index in my lattice cell number 1. If I in cell number 2, I give it cell number 2. So in a sense, this is like it's essentially taking a digital snapshot and taking it literally when we're going to represent the cells. And so something that's fundamental in a simulation environment like Computational or other modeling environments that use the same methodology, Morpheus would be an example. Artisto uses the same methodology. Chaste, which is one of the ones on our list, actually has multiple sub-packages which use different methodologies. It supports a lot of them. It has a lattice-based method too. All use the same approach. This lattice-based multi-agent approach. So once you have a cell, the next thing you have to do is define how the cells move. And the key step in each one of these methodologies is quite different. And this is really the dynamics are really the core differences between these methodologies. The way we specify behaviors is also somewhat different, but those tend to be more shareable. And so in this lattice methodology, we have a stochastic updating rule, and that stochastic updating rule may seem a little bit complicated and a little bit unnatural, but there are good reasons why we use it, both computational reasons and biological reasons. And we can talk about those. But one of the things that it's going to result in is that there are going to be some parameters that are going to be used to specify models here that might or might not exist in other methodologies. And so here's our cell, and we want the cell to move. And we're going to have a concept of what we're going to call a voxel copy attempt. This is the finest grained step inside of Computational. And this is run literally millions of times every time step inside of Computational. And so what we do is we have a state of our system, and here I've got in blue a cell in my cell lattice. I'm going to select at random a voxel, and I say voxel because these could be 3D as well as 2D, in my cell. And I'm going to ask the question, what would happen if my cell essentially protruded a little bit and extended itself to occupy a little bit more space. So here you see the red voxel is the source voxel, and I'm imagining that it's going to try to extend into that little green spot over here. And I'm going to have a set of rules that are going to say, if making this change causes the cell to get closer to the things that I've defined as targets for the cell, for example, the volume of the cell is closer to its target, the position of the cell is closer to a source of the chemical that it consumes, then I accept that update. So I basically make that move. If the move makes the situation of the cell worse, according to some set of criteria, I still may make that move, but I make it with a probability that depends on fluctuation amplitude. And that's good. I'll come back to that in a little bit more detail later. And so here I'm going the other way. In that case, I've added a voxel to my cell. And in this case, I've shown the blue has accepted that voxel attempt. Here, now I pick another voxel in red. I try to overwrite that. And so in this case, that black, which represents medium in the environment, is overwriting the blue. So the cell is shrinking a little bit. And again, I calculate how close my cell is to its targets. If making that change brings the cell closer to its targets, I accept the change. If it moves it away, I roll the dice and accept the change or not, depending on the outcome of my dice roll. And so the key thing in Computational, in any of these kinds of methodologies, is how I specify what those targets are. And in Computational, if people have done neural networks, they're used to the idea of a loss function. So, or a penalty function, which is how good your system is compared to some ideal. In Computational, you have the same thing. You have something called an effective energy, which defines essentially the degree to which the state of the system matches or doesn't match some target. And just as with loss functions in machine learning, you could have a very simple loss function, which could be, say, the pixel-wise mean squared distance between the value of each pixel in an image and the target. But you might also have something more complicated where you have that and something else and something else and something else. With Computational, each behavior or interaction that you try to specify about the cells is going to correspond to one term in this effective energy. So, for example, cells are going to have a volume. And so you're going to have an effective energy term that says my cell should have a volume of such and such. You might say that the cell has a surface area. So you're going to have an effective energy term that says the cell should have a surface area of such and such. I might say that the cell is elongated. So I measure the moment of inertia of the cell and say that the moments of inertia should be such and such. Yes, please. Yes. So typically what we will find is that there is a weighting term for each term in this, because this is getting to be a little bit more technical. And depending on your background in terms of physics or mechanical engineering, you may want to get into the details or not. If you've done this again in machine learning, it seems funny to me to be using machine learning as an example for this, because I think of machine learning as more complicated, but people typically have had it as a background. If you're doing loss functions in machine learning, you'll find that if you want to train effectively, The various loss functions, when you calculate them, have to be weighted so that each loss function has approximately the same contribution to the total loss. And so that kind of weighting becomes important. If one of them is very big compared to the others, then the learning algorithm will ignore the other ones. And so you'll see that happen here. And we'll talk about the different kinds of energy terms that are present. Within Computational, energy terms are called plugins, which is not the most beautiful name in the world, but that's how they're implemented computationally, so that's why they have that name. And why they have you'll see some oscillation in the notation for effective energy. Sometimes you'll see a curly H, sometimes you'll see a curly E. Different people use different letters for that. But the nice thing about this is that you can specify each behavior separately and add them up. So I want my cell to have a volume. Okay, I give it a volume. I want my cell to have a volume and a surface. I do both. And so I can understand those independently of each other and then in combination. When I make my update of my configuration of my cells, this effective energy will change. The volume of the cell will change. The effective energy correlated with the volume will change. The surface area of the cell will change. So there's a surface energy that will change. If there's a contact energy where cells are attached to each other, I've changed the surface area, so that will change. If the cell has a gradient which moves it, which has an energy associated with its position, that will change. And so each term in the effective energy will change when I change the lattice configuration. And that gives me a change in energy when I move that voxel in the copy. If I'm a physicist and I think about a change in energy, delta H, and a movement, delta X, delta H over delta X is a force. And so basically what I'm doing here is defining a force as the gradient of a potential energy. If you're not a physicist, don't worry about that. But if you are, that should be clear. And that's a force that acts on the boundary of the cell. And in a classical system, if I push on an object, it will move in response to the force. And if cells were purely passive, that would be all that would happen. When I push in the direction of a force, the cell moves. If the force is going the opposite direction, and somehow I'm pushing against something, and the force is unfavorable, I don't move in that direction. And the key thing about biological systems is that biological cells are active agents. They have motors in them, and they move actively. And so cells will generally move. If you push on them, they'll move in the direction you push on them. But they can also push on their environment. So even if there's a force against them, they can push uphill. Water won't flow uphill. Cells can move uphill. And so there's a probability which we define, and it has the form, again, for physicists, of what's called a Boltzmann factor, which says that if a movement is unfavorable, we make that movement with a probability that decreases exponentially with the cost of the movement, with that delta H, rescaled by a fluctuation amplitude, which has the unfortunate name of temperature. And again, temperature is inherited from physics. Biologists don't have to worry about it. Call it membrane fluctuation amplitude. And why it has an exponential form is something that we won't go into here, although if you look in the literature, there are arguments for why it has that exponential. If we want to understand how we specify what we think of as quasi-static properties, like the volume of the cell, one of the things that we have to get used to when we use these different simulation methods, and this is going to be true of Physicell in a different way. In Physicell, cells are points. They don't have a volume associated with them. there is no volume. The volume is just the fiat number that's attached to the cell. The cell is given a potential energy, which defines roughly the distance to its neighbors, and that gives rise to an effective volume of the cell. But the cell itself doesn't have a volume. In the same way with CompuCell, we can't assert the volume of the cell to begin with, because the volume of the cell is a result, the actual volume of the cell is a result of the constraints that we put on the cell and the environment the cell finds itself in. And as an example of this, let's suppose that we had a box and we have balloons and we want to put the balloons in the box. And we start out and we have a balloon that has some volume and we put it in the box and the balloon still has that volume. We now put a second balloon in the box. Those two balloons both still have their original volume. At some point, the box is full. And if we continue to stuff balloons in the box, to get those extra balloons in, they have to compress. And so the volume of each balloon will go down as I add balloons to that box. And they'll follow the ideal gas law when we compress. And so CompuCell cells actually have volume defined in the same way. Every cell is going to have a target volume, which is the volume it would have if nothing else were acting on it. And it's going to have an actual volume, V. And there's going to be a penalty function, an effective energy, which is defined by the deviation between the current volume and the target volume. And the simplest example of this, if we would think about it, if I had I don't have a rubber band, would be a spring, a classic spring. If I have a spring and I leave the spring alone, then that spring will have some length with no force acting on it. If I pull on the spring, it will get longer. If I push on the spring, it will get shorter. I have to pull or push to change the length of the spring. So with the cell volume, I have this idea that if the cell has its target volume, then there's no force acting on its surface. If the cell is bigger than its target volume, it's under tension. And if the cell is smaller than its target volume, it's under pressure. Again, thinking about a balloon is a good way to do that. Yeah. So let me repeat that for people who are online, because I don't know if you could hear. The question was, is there some maximum pressure at which the cell will burst? So in your cell growth simulation that you ran for the first homework, we didn't talk about this, but the cell's target volumes keep going up and up and up because the cells are growing at a constant rate. The target volumes are growing at a constant rate. Some of you will have done simulations in which you saw cells disappear. The cells get squeezed so much by the fact that there are other cells pushing in that they get squeezed out of existence. Now, that's probably not biologically very realistic in the sense that the material component of the cell doesn't disappear when the cell dies. When the cell bursts, the contents of the cell are distributed. So there's conservation of mass. But in fact, these kinds of things do exist in biology. In the ancient world, one way that they split rocks was to drill small holes in the rock, put dried beans in the holes, and then pour water on them. Because the hydrostatic pressure, when you add water to a dried bean, the bean will expand. The force that bean can generate is enormous. It can crack rock. Yes. Yes, but they didn't have dynamite in ancient Egypt. Yes, but yes, I mean, yes, absolutely, you're generating a force. But my point was just that what you're saying is absolutely true. My point was simply that biological forces can be quite large. We typically think for our own bodies, we're pretty soft. Most of our cells are soft. Plants, of course, have cell walls, rigid cell walls that are made out of cellulose. But a lot of the strength of the plant is because the cells in a tree are under very high hydrostatic pressure. So it's actually the water in the tree that's giving rise to a lot of its strength. And so the fact that cells are under pressure is something that is true of its bacteria are also under a lot of pressure. They have rigid cell walls as well that can be quite strong. So bacteria can be under quite a bit of hydrostatic pressure as well. Mammalian cells are pretty soft, so they tend not to be under a lot of pressure. Similarly, the surface of a cell, if we come back to our balloon analogy, the membrane of the cell is like the rubber surface of a balloon. It's under some tension. It's not usually under a lot of tension because the membranes aren't that strong, but it can be under some. Again, if you're talking about bacteria or plants, those walls are very strong. Bacterial membranes, cell membranes are not typically that strong. They typically also don't support compression. You try to push on a cell membrane, it'll crumple up. It doesn't compress. Yeah. The vertex model? Yes. So something so the membrane itself, so this is the more biological complexity, and our biologists in the room will have to should correct me. Feel free to correct me if I make a biological misstatement. It'd be easier to do if I had a whiteboard and could draw on a whiteboard more easily, but I could do at some points I probably will use the whiteboard feature on the Zoom, but I don't have my drawing tablet with me today. But in a lot of eukaryotic cells, we have what's called the actin cortex. That is a set of cytoskeletal fibers that are just roughly parallel to just under the cell membrane. And so even though the membrane itself is quite soft, those fibers can be pretty rigid. And so then they're basically coupled to the membrane and make the membrane behave as if it were a fairly rigid but solid material. And as I say, those are also active motors, so they can push and pull on things as well as provide mechanical integrity. So this idea of what's called a harmonic or quadratic constraint, which I've got here illustrated, is something that's pretty fundamental. If the energy goes as the square of the deviation, the force increases linearly with the deviation. So this is exactly what you get with a classical spring. The further away it is from the target, the bigger the force restoring the force to the target is. The combination, and here I've switched from the regular H to a script H, because it's an old slide. Yes. So that's a good question. I should probably Google that. So when the potential energy of something is defined as a square of the distance, as in the picture that I've got up here, the force, the restoring force pushing me back to my target goes linearly in the deviation. So if my volume is a little bit bigger than my target, I'll have a force that makes my volume decrease. That's essentially linear in the deviation. If I have it, so if I double my volume, the force pushing my volume down will double in return. That's how ideal gas for a balloon would work. That's how a spring works, and so on. And so remember that the force is the first derivative of the potential energy. So if the force goes like, if the energy goes like x squared, then the force goes like x. And the reason that the term for this is harmonic is because historically, when these were studied back in the 17th century, people in the late 17th, early 18th century, people looked at a spring with a weight on it, and you see oscillatory motion, sinusoidal oscillatory motion, which is called harmonic motion. And so this quadratic potential function leads to harmonic oscillation, sinusoidal oscillation. But the terminology, some of the terminology that we're going to, I tend to, again, if I'm teaching a physics audience, then I would dig into all of the classic physics terminology. If you're not coming from physics, the terminology may not be so helpful to you. What matters is the fact that the force will increase linearly. The force will act to restore the volume to its target. And the further away you are from your target volume, the bigger the restored force. That's what really we have to know. Okay. One of the results of this is with that dynamics that we went over before is that there's an effective force on the system that is in configuration space, in some rather abstract high-dimensional configuration space, which acts as the gradient of this effective energy. And the velocity with which I move, with which an object moves in this space, is proportional to the force. And that is something that is a feature in the world of cells, but would be a bug in the macroscopic world. If I wanted to model rockets moving in space, then they follow what's called Newton's law. Force is proportional to acceleration. In the world of cells, I have very high viscous damping. The fluid environment of the cell means that if I stop pushing on something, the cell stops moving immediately. And there's a famous calculation you do in a biophysics class, which says if you're a bacterium that's swimming and your little flagellum that's pushing you along stops rotating, how far do you move before you stop? And the answer is about the diameter of a nucleus of an atom. The cells essentially stop moving immediately. So the velocity of a cell in this microscopic world is actually proportional to the force on the cell, so you don't have acceleration proportional to force, you have velocity proportional to force. And this funny dynamics that we've got of picking sites, rolling the dice and determining whether we advance or retract boundaries gives us this result that the velocity is proportional to the force. It happens that numerically that's absolutely stable. So from a computational point of view, it's nice because we don't have to worry about the numerical stability of our algorithms. Okay, so having said that, let's imagine that we have two cells here. Here's cell number 1, and here's cell number 2. And which of these would have a bigger fluctuation amplitude? The one on the left or the one on the right? How about getting hands up? Give me a thumbs up if you think that the cell on the left has a higher fluctuation amplitude. Remember that the fluctuation amplitude gives me a higher probability of something unfavorable to happen. So both of these cells are starting in the lower left and they're crawling to the upper right. So something is pushing on them. They both have approximately constant volume. But do I see more fluctuations in one than the other? Which one has more fluctuations? The left one or the right one? The one on the right. So that's telling us that the fluctuation amplitude for that cell is high. And if I look at the one on the right, it's actually falling apart into multiple clusters, which is not something that's biologically realistic. So that cell has fluctuations that are so big that it's behaving in a non-physical way. Go ahead. Yes, question. So the question for people who are online is, in the context of Computational, would the simulation break when the cell disperses into multiple clusters, or does it stay one cell in quotation marks? Computational defines a cell as a collection of voxels which have the same cell index. And therefore, even if the cell is not composed of connected voxels, Computational will treat that as a single cell. And that can have some perverse results in Computational. It's not easy, but occasionally somebody will write a simulation where you do something where the cell will split into two pieces. And you'll be doing something to the center of mass of the cell. And the center of mass will move the way you want it to, but the two pieces will merrily go off and do things that don't look like they should be doing it, because there's nothing that says that the cell has to remain connected. And there actually is an option in Computational to say that cells have to maintain connectivity, that they're not allowed to split apart. That's an option that you can impose. Typically, we don't do that because actually checking connectivity is a fairly computationally expensive thing to do. So it slows the simulations down. If you design your simulation well, you shouldn't need to do that, but there are times when you do. And so this is an example of a pathology that you could get in the simulation methodology. Great question. Other questions or comments at the moment? All right. So here, that was a single cell. Now we're going to look at a cluster of cells. And we have two clusters of cells. And these cells are interacting. All the cells have the same target volume. They're interacting through contact energies, through basically the adhesivity between cells of different types. And what you see is that the blue cells are coalescing to make a cluster, which means that they cohere to each other more than the green cells. And we'll do that simulation in a little bit. But you see that the shapes of the cells are a bit different. And the question here is, which one has a bigger fluctuation amplitude? And if you see on the right, the boundaries between these blue cells have basically fallen apart. The blue cells are basically mixing with each other because the fluctuations are so big. And so that's another pathology that one has to watch out for in this kind of simulation. You'll see something else, which is one of the things that you face in Computational is that normally we define the fluctuation amplitude for the simulation as a whole, although you can in fact define it by cell by cell. And so you'll notice that the green cells here on the right, the boundaries are squared off and rather rigid. And so effectively, this is a simulation on the right where the fluctuation amplitude is too small for the green cells to behave right, and it's too big for the blue cells. And we'll come back to that in a few minutes when we start running with simulation. So we're seeing some pathologies of these simulations already. I'm not sure, this is a slightly different set of slides from the ones that I've used in previous years. You'll see these are credited to Giulio Belmonte at North Carolina State, who teaches this method and uses it a lot. Yes, go ahead. So yes, so the question was, is the reason that the cells are squared off because we're operating on a square lattice? The answer is yes. One thing Computational lets you do is run on a hexagonal lattice, which is a little bit better behaved. And you can also change the lattice interactions to try to reduce lattice effects. But being on a lattice has certain advantages computationally. It makes computation much simpler, much faster than going off lattice. But it does mean that there are lattice effects. The universe is not on a square lattice. And so things like measuring distance are a little strange on lattice. Yes. You mean, how do we actually implement hexagonal and square lattices computationally? So effectively, you don't lose much by going to a hexagonal lattice. The way lattices are implemented is, again, we're jumping into sort of technical details, but it's a very reasonable question, especially in a computing course like this. It's a very reasonable question. When you do these kinds of simulations, we talked about picking a lattice site and then picking a neighboring lattice site. And so the software, and this is actually one of the things that makes writing code extensions for Computational a little bit cumbersome compared to doing things for hard-coded packages. Computational allows you to define lattices that are 2D or 3D, square or hexagonal, and also an arbitrary interaction rate. You could say my nearest neighbor is second neighbor is third neighbor is fourth neighbor is fifth neighbor, and it will handle that all for you automatically. And so actually, one of the nice things about hexagonal lattices, technically, is that a hexagonal square lattice is a square lattice, but you've defined different adjacency rules for it. So if I define a square array, and I say my neighborhoods are my normal square arrays plus 2 additional neighbors, that turns a square array into a hexagonal array. If I do the same thing in 3D, I get a hexagonal lattice in 3D. And so I don't have to actually build a data type that is different to represent a hexagonal lattice. There are other kinds of lattices that I might want to use where I actually need a different data type, which is a pain. But for hexagonal lattices in 2D and 3D, I could just use square arrays. I don't have to do anything fancy. No. Okay. Okay, so the effective energy change, delta H, for a typical voxel copy, divided by that fluctuation amplitude determines how big our fluctuations are. If delta H is small compared to T, then my fluctuations dominate, and I'm like that left hand, the second one over, where my cells are falling apart, or the blue one here, where my cells are dissolving. If T is too small compared to delta H, then I get rigid and things freeze. And again, if you're a physicist, effectively, you're used to a concept of phase transitions. I have a solid phase, which is when things are lined up on the lattice. I'm frozen, essentially. I have a liquid phase, which is what I want, which is intermediate temperature. And then if my fluctuations are too big, I get a gas, things fall apart. And so effectively, you're actually seeing that here in the one on the top right. The green cells are effectively frozen, they're solidified. The blue cells are gaseous. And what I want is something like the leftmost picture, or the cluster of cells on the left-hand side, where the cells have fluctuations, they can move relative to each other, but they're staying as a compact object. And so these are situations which we have to think about when we're designing simulations, because Ultimately, most of these kinds of things that we're seeing here are artifacts of the simulation methodology, not reflecting the biology. And ideally, in a simulation method, we wouldn't have to think about pathologies right away. And sometimes we don't. Sometimes we just go ahead and start simulating things and then come back when we've had the pathologies. But my experience, giving you a heads up about what you may run into early isn't a bad thing. It'll make more sense when you start playing. But over the years, our experience is that people will go into a parameter regime where cells behave strangely and not know what to do. Or maybe not even recognize that there is a problem. The simulation will break in some way. And so I'm giving you a little bit of a heads up about what some things you may wind up with. Some of these behaviors you'll actually see. In other words, this kind of frozen behavior that you're seeing in those green cells is typical in plants. And in fruit flies, there also is very little relative cell motility. Cells have relatively rigid boundaries. And so it's not that these things can't be relevant biologically, but you want to be choosing them rather than having them be an accident because of the way you set up your simulation. Other questions? Those are great questions. Okay. See if I can get to the next slide. Okay, one of the things that we are going to run into in Computational is a question of what timescales are. You'll see the rather mysterious term Monte Carlo steps used a lot in simulations. And what is one Monte Carlo step? One Monte Carlo step consists of, if my lattice has, say, 1,000 by 1,000 voxels in it, I have 1 million lattice sites. If I try to do 1 million voxel copies, that is one time step. That is the natural time step defined in the simulation. A cell can move at most 1 voxel per Monte Carlo step. Actually, because the movement is stochastic, a single cell could move maybe a little bit more, a little bit less, because I could accidentally pick the same cell more times than I should. Because I don't guarantee that I visit every voxel once. I randomly pick voxels. I could have a different algorithm where I have a list of all the voxels and I guarantee I visit each one once. That would actually be better behaved, but it's computationally quite expensive to do that calculation. So we make the approximation of doing random picks. And actually, the single thing that's most important in this kind of particular algorithm that we're using is the quality of the random number generator that's doing the picking. If you use a crappy random number generator, you see pathologies really well. It's actually an extremely good test of the quality of your random number generator. These days, that's not so much of an issue because most random number generators are pretty good. But back when I started out, the default random number generators in Fortran or C were really crummy. And if you tried to use those default random number generators, pseudo random number generators in these simulations, they didn't work. So use better pseudo random number generators. Well, they're published algorithms. Well, you don't have to do that. These days, you go to Stack Overflow and look it up. But in those days, there was a wonderful book called Numerical Recipes by Vetterling and Press, which was from Harvard. It was from some astrophysicists at Harvard, published this book of numerical methodologies. I don't think people use it so much anymore, but back 35 years ago when I was a student, that was the Bible to go to if you needed to do numerical computation. They had beautiful, very well validated, very well designed basic algorithms. That's the pre-Google days. So depending on what you're doing, the quality of your random number generator may or may not be critical. And the better the random number, pseudo random number generator you use, the slower it is. And so the algorithms that are used in the old compilers are very, very fast, but their quality is pretty low. And so that's the trade-off. I mean, these days, a lot of CPUs actually have true random number generators, where they use quantum or thermal noise to generate random numbers that are true random numbers. The problem with those is, one, they're fairly slow, and two, you can't reproduce your simulation, because if you use a pseudo-random number generator, you can set the seed for the random number generator, and then if you use the same seed twice, you'll get exactly the same answer. If you use a true hardware random number generator, then every time you run it, it will be different. There is no way to replicate. In practice, what happens is that if you want true random numbers, what they'll do is they'll pick a true random seed from the hardware generator, use that as the seed for a pseudo-random number generator, run some number of time steps, and then pull another one, because doing it every time is too slow. But almost certainly, if people have current CPUs, your computer almost certainly has a true random number generator hiding somewhere inside of it. Computational doesn't use that, but you probably have access to it through Python if you wanted. You'd have to look. That access is different on different CPUs because it's a hardware dependent thing. Okay. So the comment was, well, why don't operating systems handle true random numbers differently? I mean, equivalently. The answer is that they don't. Good. That's a good topic for over drinks, I think. There are a lot of those. So it's about the hardware. And for the biologists in the room, please don't get too intimidated by the computing talk. We'll have plenty of biology talk where we can talk about genes and intimidate the computing people. Okay? We can find it. We can find a collective medium between all of the different languages. Okay. All right. But there is this issue that there's a certain Computational is what's called the speed limit, which is if I push things too fast, they break. But at the other level, I can define this natural time step to represent any time that I want that's shorter than this limiting time. And so typically, the time scale wall clock, the time represented in real time compared to Monte Carlo steps is defined by the fastest moving thing in your real world. So if you have cells that move 1 micron per minute, and my voxel size is 1 micron, then my Monte Carlo step has to be less than a minute. If my cells move 1 micron per 10 minutes, and my voxel size is 10 microns, then my Monte Carlo step could represent 10 minutes. And so, or 100 minutes even. So you have to think about what basically the time equivalences are defined by whatever the fastest moving thing in your simulation is going to be. In living tissues, typically cells don't move very, very fast. They don't crawl around very fast. And so that means we can get away simulating things where our time steps are minutes or tens of minutes. If I'm modeling immune cells, unfortunately, immune cells crawl around pretty fast. So that means that I have to use smaller time steps. And if I'm modeling something like blood flow to capillary, which is pretty fast, then I wind up having to do very short time steps in my simulation. And that slows things down. So those are trade-offs that we have to play with. Okay. Computationally, Computational and most of these other packages that you'll face do not do very fancy interfacing between different algorithms. They just alternate time stepping of different algorithms. So in Computational, you will do one lattice update where you move the cells around. Then you will do something where you update all the chemical fields, for example. And then you'll move the cells around. Then you'll update the chemical fields. Then you'll update the network state inside of the cells. And so you do time slicing where you alternate between different solvers. That can cause numerical instabilities because you have stale parameters, if you're interested in that kind of thing. And Computational allows you to do some, there was a question actually last week about can you actually control the granularity of those steps? And the answer is yes. It's a little clunky to do it, but you have some features that allow you to change that. Because these other solvers are run between Monte Carlo steps, the computation has had the rather ungainly name of a steppable. And you'll see in Computational steppables loaded. Those are alternated with the movement of cells. So you move the cells, you run the steppables, you move the cells, you run the steppables. Okay, so now let's actually do some coding. And we're going to start out by trying to understand how to control cell shapes and behaviors based on these simple differentiation that we've seen here between cells that have too much fluctuation on the left or not enough fluctuation on the left and cells that are behaving like cells on the right. We're going to be thinking about this in the context of biology and the difference between epithelial cells and mesenchymal cells. As always in biology, the language tends to dichotomize things. We tend to say apoptosis versus necrosis for cell death. We tend to say epithelial versus mesenchymal cells for tissue organization. Those limits are well defined. If we see true epithelial cells or true mesenchymal cells, we know they're different. But in fact, there's a continuum of intermediate states between them. And you'll see names like epithelioid for cells that are sort of somewhere between mesenchymal and epithelial. But basically, epithelial cells are cells that are typically in sheets. They don't move around a lot, although they can move. They're polarized. They have apical and basal surfaces that are different. And they often are joined to their neighbors by junctional adhesion. Mesenchymal cells tend to be, if not round, because some very complex cell shapes like neurons are mesenchymal, but they don't form sheets. They tend to be more typical of three-dimensional organization. There always are going to be some counterexamples in biology. And if you're dealing with bacteria, then they have a totally different structural organization from what we're talking about here, or plants. But we're going to imagine that we have this concept of epithelial sheets where the cells are all connected to each other versus mesenchyme where the cells are more round and more fluctuating and have some space around them. And we're going to imagine epithelial sheet. We're going to imagine that we're going to do this in 2D. So we're going to look from the top. An epithelial sheet, all of our cells would be attached to each other and stuck and not moving around a lot. Mesenchyme, we would expect the cells to be surrounded by medium and also to move more freely. And so now let's actually go back and create a basic simulation. And I want people to do this. I think for the sake of the room, I'd rather not do it as a split screen. Should I do it? Would people prefer that I do this in real time as we go through it? Or is it okay to show you the slides and have you do it and tell me when you're okay with it? I'd like people to create a basic simulation. So if you're on nanoHUB, go to Twit it. If you're local, go to Twit it. And create a new project. So we're going to go, maybe I should, what do you think, Brianna? Should I do this? Should I split screen it? Okay. Okay, so let's do this. Give me a second to redo my screen layout a little bit. There seems to be an issue with the resizing of the The problem is that there seems to be an issue with the resizing of the slides. If I try to compress the slides too much, they don't compress right. Okay. Okay, does that work for people? Can people see that all right? Our online people? So what I want people to do is I want you to go to Quit, which is what I have running here on the right, and go to CC3D project, do CC3D project. Okay, does that work for people? We did this last week, but we'll walk through it. Okay, and we're going to create a new module, and here I've called it module 1.3.emt, but let's just call it. I'll just call it EMT for the moment. Remember, when you create a simulation name, don't give it don't start it with a number, and don't use special characters except underscores in your name, especially if you're on nanoHUB. Okay, so we're going to create a new simulation. Next. Okay. And now we're going to basically accept all of the defaults for the lattice size. Now that you've seen that we're on a cell lattice, you can understand perhaps why there's a lattice size in this simulation. There's also this term pixel copy range neighbor order, which defines a little bit of how the lattice works. There should probably be a button here that says hexagonal versus square lattice, but we don't have that. That should probably get added to this. Oh, I'm sorry. Yes, it is. Sorry. So lattice, you could say square versus hexagonal lattice. That was that. My mistake. So if you want to run it on a hex lattice, you got it. That was something we didn't have. I am often out of date because I've used this code for so long. I don't always catch up on additions to it. Students often point out to me that I'm doing something in a difficult way because it didn't exist when I started doing it 10 years ago. Okay, so we'll stick with a hex square lattice for the moment. You can play it. We're going to start out with a square of cell, so we'll hit rectangular slab. Next. And we're only going to create one kind of cell. So we'll call it, example here was just my cell. I don't know why he picked that. My cell. Add. So if I'm going too fast, speak up, please. Everybody try to keep up with this. So we're going to create just one kind of cell. Everybody okay with that? If people need more time, speak up. So freeze would say that the cells do not move. So if I want to build something like a rigid wall that represents bone or the glass that my cells are sitting on, I would hit freeze. That gives me a wall that doesn't ever move. Okay, so I have one cell type. I'm not going to create any chemicals, so I hit next, skip those. And now what am I going to do with my cells? I'm going to give them a contact energy. I click contact. I'm going to give them a volume. And to begin with, that's all I'm going to do. I'm going to give the cells a volume and a contact energy. So click contact, volume flex, center of mass is clicked by default. So I don't have to do it. Okay. And now I hit next. And I hit finish. And I had that other more complicated simulation running, but I have this simulation here. And now I have that simulation defined, and I'm going to right click on its name and I'll say open in player. And if I'm running desktop, it will just open. If I'm on nanoHUB, it will start a new simulation. And you should see something that looks like that. Your colors may vary. Did people get that? Anybody stuck? Are online people all right? Everybody okay? So let's run this simulation. I'm going to run it. I see the cells move a little bit. You can lay things out on your computer so that they're prettier. You can have things be bigger and so on. And I can zoom in. Let me zoom in. And actually, whatever the default parameters are for this is not too bad. If I look at it, I don't see the cells falling apart. They're not falling apart into lots of little independent voxels. And the boundaries are not all lined up with the lattice. They're not on squares. So the default parameters that I picked are pretty good. If I look down here in the console window, it'll say that there are 484 cells in my simulation. Your number may not be quite the same. You'll see something called step. That's the Monte Carlo step of the simulation. You'll see something called flips. Flips is the number of accepted copy attempts per Monte Carlo step. Remember, we had this idea, we try to make a lattice copy. We may make it or not. That's how many times we actually made a copy. So if that number is very small, our simulation is basically frozen. If that number is huge, we're basically making random updates. What's a reasonable number is something we have to think about. There's a term, something called energy, which is that effective energy that we've calculated for the simulation. And you'll see that number bouncing around a lot. It's not actually by itself a very useful number. And then we have number of cells in inventory. And the reason there are two of those is a bit obscure because they really are providing the same information. Okay, does everybody have that working? Does anybody need time or help getting that running? So there's a comment, fluctuation is something similar to resistance. Yeah, 1 over resistance, perfect. Yeah. Okay, and remember your buttons, pause, step, play. And if you hit stop, you have to start from the beginning. So don't hit stop unless you absolutely have to. And one thing that you'll find, at least on nanoHUB, is when you hit stop, the screens will all resize and then you'll have to relay them out. So that's a bit of an annoyance. Okay. One simple thing is that the color choices that you get may be different. I don't know what color your simulations were. This button here, which you can hover over, called configuration, allows you to do things like change your color. So here my cells are purple. If I want my cells to be yellow, I click on yellow, click OK, OK, and now I have yellow cells. If I want the boundaries between the cells to be a different color, I can say cell border color here, black. I don't really know why I'd want it to be red, but I can make it red. See if that works. Didn't pick up, I don't think. I have to try that. Try that again. Cell border color, red. Yeah. Didn't pick it up. Does that not work? No, it doesn't pick it up. Yeah, doesn't let me update it. Oh, I see. There's a saturation number. Thank you. It's too fancy. Okay, now I've got red down. Thank you. Another feature that didn't exist the last time I tried to play that. So you can change colors. That's not, I mean, not a big deal, but occasionally you'll get some reasonably offensive color schemes, which are you may want to be able to play with. So if I start the simulation over again, I'm going to look at the initial configuration. And the initial configuration is a bunch of squares, which is not very natural. But it's easy to draw. And if I step this, what you'll see is that the boundaries move a little bit at a time. In each step, you'll see my boundaries are joggled by 1 pixel. You look at that carefully as you run. My boundaries joggle 1 pixel at a time. And you notice that the boundaries move basically less than 1 pixel per Monte Carlo step as I do step. If I hit run over a long time, the initial squares will fall apart and I'll get something that looks more hexagonal, but it takes a while for that to happen. The reason for this shaping, and I have 2 shapes, one of which is the boundary between individual cells within my cluster, and the second one is the shape of the cluster as a whole with respect to the medium. Those are 2 time scales, one of which is a local rearrangement within the cluster, and one of which is the rounding of the cluster as the droplet. The boundaries within the cluster change rather rapidly. The rounding of the cluster takes quite a while for this particular set of parameters. So that's something, and these are all due effectively to the contact energies that we've defined, because that's all we've got in the simulation. Or actually, to be more specific, the ratio of the contact energy to the fluctuation energy. Okay, so let's talk. We only have 10 more minutes, so let's try to talk about contact energy. One of the most important things that cells do is stick to each other. My old friends Gabor Forgax and Stuart Newman wrote a beautiful textbook on the physics of biological development. And the very first sentence in the book is that the most important things that cells do is stick to each other, because otherwise we'd all be single cell organisms. I suppose one could make an argument that there are other things that are important about biology as well as cells sticking to each other, but it is true that if cells did not stick to each other, we would have lots of bacteria, probably, not anything much more than that. Now, of course, bacteria could actually stick to each other and build quite complex structures, so it's unfair to bacteria to say that they only behave as single suborganisms, but that's a different thing. So one of the things that we will do in Computational, and this is a little bit artificial, and Computational allows you to do these things in a more biologically meaningful way, is to define cell-cell adhesion. When you have 2 cells or cells and medium that are in contact with each other, we have to define the energy of that contact. And you already played with that a little bit in the homework, but we hadn't really explained the concept. And so now we're going to come back based on what you play. This was an example where we play with it a little bit, and then that hopefully motivate why we want to talk about it when we get to class. And so what we're going to define is biologically, contact energies are determined between cells primarily by cell adhesion molecules that are expressed on the surfaces of the cells. In particular, we're going to have things like cadherins that are cell-cell adhesion molecules, or integrins that define the energy of contact between cells and the extracellular matrix around. To begin with, though, we're going to ignore the molecular complexity, and we're going to simply say that cells have some typical energy per unit area when they touch each other or when they touch medium. And so we're going to be able to represent that as a table that says the energy per unit area of the surface of the cell between two cells that are of type 1 or 2 or 3 or between cell and medium is something. And that, when we have only a couple of cell types, gives us a very compact way of specifying cell adhesion. The problem with it is that when we have a lot of cell types, that table gets very large, 1. And 2, if we want the cells to actually vary cell by cell as opposed by type, we can't use that kind of table formula. And so there'll be something called adhesion flex that we'll deal with later that allows us to specify cell-cell adhesion by molecular species rather than by cell type. If you're interested in the mathematics, there's a simple formula. And if you're really a physicist, this formula is basically the Ising model, which says that if I have two cell voxels that are at the boundary of a cell, they contribute an energy that is given by the identity of the cells on either side. Then we sum that up. You can think about this as a surface energy. If I have a cell that's by itself and it has some surface energy per unit area, there's an energy of contact between the cell and the medium, then the total surface energy of the cell is just the contact area of the cell times with the medium times the energy per unit contact area. When you're on a lattice, things are a little bit more complicated because you have to sum up over lattice sites. And that means that there is a parameter, which is the range over which you do that evaluation, which makes things a little bit more complicated. For our purposes, it's not going to be that critical at the moment. In the case that I have here, we only have two cell types. We always have a default cell type, which is medium, which is where I don't have cells. And then we're going to have our single cell type, which we call my cell. And that means that I have in principle 3 possible contact energies. The energy between medium and medium, which actually doesn't do anything because I only have one medium cell. The energy between cell and medium, which is the energy of the boundary of the cell of this aggregate, and the energy between cells and cells. And so there really are only two energies that matter: cell medium energy and cell energy. Okay. So one of the things that we could do is we could change the contact energy between medium and cells. And we could do that in two ways, and we're going to run out of time in a second here. But I want to show you how to do it anyway. We're going to go to our XML. So we go back to our simulation, our Twit. And in Twit, we are going to scroll down. I have to change my font. In Twit, we're going to scroll down, and we are going to see here in lines 36 through 42, it says plugin. Remember, energies are called plugins in Computational. Contact energy. And we'll see a series of lines medium, medium, and then a number. Medium, my cell, and then a number. My cell and my cell, and then a number. And then a neighbor order, which says the range over which we do this calculation. And so let us change cell medium energy from 10 to 20. And I'll hit save all here after I've made the change. So that's what the code looks like. It's expanded a little bit. I'm going to make that change from 10 to 20. Everybody do that. I'll hit save all. Sorry, save all is the double floppy disk icon. So change cell medium cell to 20. And now if I go back to player, whether it's local or online, I hit stop and I run again. And provided that things are behaving themselves, that may be a big if here. If on nanoHUB, if the screen doesn't redraw, joggle the bottom corner here, and it will force to redraw the screen. And you will now see the simulation running with the higher cell medium contact. The difference here is pretty subtle between these two situations. The other thing you could do, and this is the last thing I want to show you, is that you don't have to change the values in the code itself. A lot of the values can be changed while the simulation is running, which is very convenient. In this box in player called Model Editor, if I open up plug-in contact here, one thing I have to do is I have to open things up by hand. I will see energies listed here. And I see type 1, type 2, my cell, medium, my cell, and the value, 20. And so I could go in and I could change cell medium contact energy, make it 5.0. And one thing that's a little bit of an annoyance is that if the thing was initially a float, you have to make it a float. And if it's initially an integer, you have to make it an integer. So if it was 5.0 and you type 5, it won't work. And I hit enter. And I've now reduced the contact energy between cells and medium. And so the cells have a lower energy with medium, they'll begin to disperse into the medium. 5 happens to be neutral. Try making that value 3. 3.0. Now my cells will migrate out into the medium. And so this is a very, very simple simulation of what's called epithelial to mesenchymal transition. I've gone from a situation where the cells want to stick together and form a cluster to a situation where the cells want to disperse in the medium. Now, in reality, for example, when epithelial mesenchymal transition happens in cancer or in normal development, or similarly, mesenchymal epithelial transition happens, a lot of things change in the cells beyond the cell-cell adhesion. But changes in cell-cell adhesion are, in fact, critical to the metastasis of cancers. And they're critical to processes in development. During development, cells often go between sheets or compact clusters and dispersed migrating groups of cells, and they go back and forth many times. And so during normal development and also during tissue maintenance, you'll see situations in which cells go back and forth between cohering to each other and forming a coherent cluster and dispersing in medium. And so this is a tiny simulation where we can simulate one very simple aspect of that. So in our last -3 minutes, why don't people try changing those two parameter values, the cell medium by cell contact energy, and if you like, you could also change the by cell contact energy. One thing that you'll find, which you have to think about a little bit, is that contact energies go backwards from what we might expect. Higher contact energies mean you're less cohesive. A bigger energy pushes you away rather than it attracts you. And so if you want things to stick more, you use a smaller number rather than a bigger number. It's also legal to make that number negative, to actually have things in here. But if you try that here in this simulation, you'll get something pathological. I can try it. The simulation did exactly what I asked it to do. I say that the contact energy between cell and medium is -3. That means the more contact area I have, the lower the energy of my simulation. And so that means my cells fall apart into dust if I try to use a negative contact energy. If I tell the cells that they can't fall into dust, that they have a fixed surface area, then that would work. So if I actually want to use negative contact energies, I actually have to define that the membrane area of the cell is finite. What I've done here is I've used the contact energy to minimize the surface area of the cell and its configuration at the same time. That's why that doesn't work. And so I hope that that gives you something to play with. There are a bunch of exercises that we could do here. And in the homework, you'll explore these. I have in the slide deck, I have a bunch more of them. And you'll have access to that deck if you want to explore them on your own. But if you can make this change, the simple change by editing these energies by yourself, you'll be able to do all of the simulations that come below. Okay. Are there any other questions or comments before we break for today? Go ahead. Okay, so the question was, how do we change the temperature? How do we change the fluctuation amplitude in the simulation? So in the editor, if I go to the top of my XML, at the very top, I will see this line 16, which says temperature. And by default, that's 10. I do not remember, this is something we can try together. If you go to plug in, if you go into the model editor, at the very top where it says pots, it says temperature here. And in principle, I can go in and I can change that number. I do not know if it will actually update it or not. So let's try it. I'm going to go into my model editor. I'll change the fluctuations from 10 to 1. That should freeze. If that works, all my cells will freeze. And they do. So you could change it on the fly as well. It's possible to define different fluctuation amplitudes for different cell types, but that has some subtleties to it that we're not going to get to for a while. If I multiplied my temperature, if I divided my temperature by 10, the way I just did, and then I divided all of my contact energies by 10 and my volume energies by 10 and everything else, then I get exactly the same simulation, because it's only the ratio of the effective energies to the temperature that matters. Other questions? I realized this was rather a codey and mathy lecture, not a very bio-y lecture. So I hope for the biologists in the room that wasn't too traumatic. I promise you that there will be plenty of lectures which will be much more biologically focused. And I hope that gives people a place to start. I think looking at the homework, you should have everything you need to be able to do the homework assignment for this week. Again, if there's a homework assignment and you look at it and you say, I have no idea what you're talking about in the homework assignment. Step 1, look at the Computational website and the manuals and see if you can figure it out. Don't spend a day trying to figure it out, but spend 15 minutes trying to figure it out. If after 15 minutes of digging on the Computational manuals and demos, you can't find the answer, then play a little bit. And if after 15 more minutes you can't figure it out, then write and say, I'm going to do this problem next week after we've explained it in class, okay? Or bring it up as a question. Give yourself a little bit of a push. Don't. If it's something unfamiliar, don't just stop and say we haven't done it. But don't kill yourself trying to figure it out. Is that a fair balance between the? Again, because my experience is that if you've played with things a little bit, then when we cover them in class, they'll seem more motivated. They can often seem pretty abstract. If I introduce a concept like fluctuation amplitude, you haven't played with it. Where does that come from? Now you've seen it, now we can play with it. And then we can talk about it more and come back to it. All right, so I realize that can sometimes be that you're feeling a little bit flying blind on the homework. And so I don't want people to feel nervous about that. It's okay. If it doesn't make sense, just say so. You always have the opportunity to come back. Okay, anything from our online people? Final opportunity for questions before we break. Did the videos and things work for our online people all right? I know the screen shares and things aren't ideal, but I hope that people could see all right. Okay, well, I want to thank everybody for coming, and I look forward to seeing you next week. And